All right, this video is the last section for Chapter 6, Section 3, which covers cellular respiration. This fermentation lecture is pretty much what happens to cells when oxygen is not available anymore. So when there are two different types of conditions for cellular respiration, one of them is when there is oxygen and when there's not oxygen. Okay, so hopefully we saw with the end of the last lecture that oxygen, when available, becomes the final electron acceptor, and that's where we have the formation of water at the end of the electron transport chain. But under anaerobic conditions, okay, the NADH, which is a high energy molecule, it gets oxidized by a process of fermentation. Okay, so there are many different types of fermentation, okay, but they all operate to regenerate the low energy portion of NAD, because then that NAD will go back into the beginning of glycolysis and get reduced. Okay, so the overall yield of ATP is much lower than during aerobic respiration, or the respiration with oxygen. There's only 2 ATP formed, and that's just the 2 ATP that are made net during glycolysis. Okay? So one type of fermentation is lactic acid fermentation. So in this case, the end product is lactic acid. And the NADH that was formed during glycolysis is now, instead of going to be used in the electron transport chain to transfer high energy electrons to push the protons across the concentration gradient in the mitochondria, we're going to have it, and it's going to reduce the pyruvate into lactic acid. So a couple things are happening right here. The NADH during anaerobic processes is not going to be used in electron transport chain, because oxygen is going to be needed for the electron transport chain. And then also the pyruvate, which is during aerobic respiration, is converted into acetyl-CoA from the reaction with the coenzyme A between glycolysis and the citric acid cycle, or Krebs cycle. But now the NADH is the high energy electrons there are going to be used to reduce the pyruvate to lactic acid, and then that of course will regenerate the NAD+, which goes back into the beginning of glycolysis and gets recharged again when the substance, or whatever the organism, splits that sugar during glycolysis. Now, unique thing about lactic acid fermentation, if oxygen ends up becoming replenished and there's enough oxygen back in the cells, then the pyruvate can be reformed when the lactate or the lactic acid is oxidized by the NAD+. Okay, so here's a picture of the lactic acid fermentation. As you can see in the glycolysis, it looks very similar. But instead of the pyruvate going into the pyruvate oxidation stage, it's going to be converted into lactic acid. Okay, and then of course during that process, that NADH that was made during the splitting of glucose to make the pyruvate, those high energy NADHs are then being used up to form the lactic acid. And that, of course, will reload the NAD pluses to go back up into the first arrow in the first stage. Again, that first arrow, that first process, is where we're getting the only ATP during fermentation. Okay, so lactic acid fermentation, glucose splits, makes 2 ATP, makes 2 NADHs, just like we do normally. Okay, but the second step, the pyruvate, is not going to be converted into acetyl-CoA to go into the Krebs cycle. Instead, it's going to be oxidized, or the NADH is going to be oxidized, and it's going to reduce the pyruvate into lactic acid. Okay? So the other type of fermentation is alcoholic fermentation. Now, the difference is the end product. Here, the end product is ethyl alcohol, or ethanol. So pyruvate is converted into acetaldehyde, and CO2 is released. The NADH is used to reduce the acetaldehyde to ethanol, and therefore the NAD+ is regenerated again to go back into glycolysis. Now, the one difference here is the CO2 does not, because the CO2 gets lost and that carbon source gets lost, there is no reformation back into pyruvate if oxygen becomes available again. So, taking a look here, Looks very similar. However, after the production of the 2 ATP, again 2 ATP during fermentation, and the production of the 2 NADHs, the pyruvate is then going to be converted into acetaldehyde, and when that happens, 2 carbon dioxides are released, and the loss of that carbon is important because it's going to prevent us from, or prevent an organism from reconverting the ethanol or the acetaldehyde back into pyruvate. Okay. Well, when the pyruvate is converted into the acetaldehyde, and then the NADH, or the NADH that was made from the first step of glycolysis, is then going to be oxidized, and that energy is going to go into converting the acetaldehyde into ethanol. Okay? And then, of course, the NADH, or the NAD plus, is then going to go back up into the first step, or the first arrow of glycolysis, and then be reduced when the glucose is split. Okay? And that is fermentation.